So, secondary structural elements in protein structure are dominated by two main motifs, alpha helices and then beta strands that interact to form beta sheets. So we're going to talk about some of the structural details with each of these. So on the left here, we've got secondary structural element, alpha helices. And on the right, we've got beta strands that interact to form beta sheets. So you notice I keep saying beta strands interacting to form beta sheets. And those are two terms that unfortunately are sort of used interchangeably, but beta strands represent sort of the backbone structure that we're going to have that make beta sheets, but two or more beta strands need to interact to make a beta sheet. So we'll get to that in just a minute. So highlighting over on the left here are alpha helices, and then over on the right are beta strands that interact to make beta sheets. So let's talk first, I'm going to zoom in on the bottom here, about a common intermolecular force that we see with both alpha helices and beta strands interacting to form beta sheets, and that's hydrogen bonding. So hydrogen bonding that occurs between backbone atoms is going to form the core of the stability of both of these structural elements. So we have a carbonyl and then a hydrogen bonded to a nitrogen, and this is the basis behind the hydrogen bonding that we see in both of these structural elements. So this is a common piece. Hydrogen bonding between the oxygen of the carbonyl containing functional group and then the hydrogen of the nitrogen. And that makes the carbonyl oxygen, the hydrogen bond acceptor, and then the hydrogen of the nitrogen, the hydrogen bond donor. So the hydrogen bond donor donates the hydrogen, and then the hydrogen bond acceptor is a nitrogen oxygen. Fluorine counts, but we don't see that generally biochemically. So nitrogen or oxygen atom whose lone pair accepts that interaction with the hydrogen to form our hydrogen bond. Okay, so that's a common piece that we see here with both of these secondary structural elements. They are stabilized by hydrogen bonding between those specific backbone atoms. And remember, we've mentioned before, structure formation takes into account two pieces. We have sterics that we need to consider. We need to make sure that...
I didn't get that. Could you try again?
Siri constantly thinks I'm trying to talk, but I'm not. So we have sterics. We need to make sure that atoms aren't banging into one another. And then we have energetics. Structure formation is entropically unfavorable, so it must be compensated for by some form of enthalpy. That enthalpy comes in the form of intermolecular forces, and specifically for secondary structural elements, it's hydrogen bonding between backbone atoms. I feel like I've probably said that 1,000 times, but it is a super important concept to get. Okay, so let's move over to the alpha helix world. Okay, so I have some models here to sort of demonstrate what we want to think about with alpha helices. So when we think about alpha helices, we are thinking about a coiled structure. So I want you to think about coils when we talk about alpha helices. So let's zoom in over here and we'll talk about some of the structural features that we see here. Remember, when we're talking about structure in peptides, we have directionality to the peptide backbone, and we want to think about NC-alpha carbonyl, NC-alpha carbonyl. So nitrogen is always blue, so here we see a nitrogen, C-alpha, and then here's our carbonyl. Okay, so NC-alpha carbonyl, NC-alpha carbonyl, NC-alpha carbonyl. Oops, there's a carbonyl. So when we think about directionality, we can see that for this alpha helix, the directionality goes this way. Now, one of the things that is important to think about is the majority of alpha helices that we're going to see are right-handed. So here's how you could figure out that it is a right-handed alpha helix. If we look at this and I point my thumb in the N to C direction, I can see that my hand would coil around the curvature of that alpha helix. So that's confirming that it is a right-handed alpha helix. I don't know how this is showing up on the video, but this is my right hand. So a right-handed alpha helix. Okay. Left-handed alpha helix is if I were to put my left hand and use my left hand and point my thumb in the N to C direction, try to see that you do not coil around the helix here. It's the opposite. Okay. So right hand is going to allow me to coil that alpha helix. So this is confirmation that this is a right-handed alpha helix. Okay. So here's our little alpha helix here. So we can see the hydrogen bonding that we have between our backbone atoms. And zooming in just a little bit further here, here again is our carbonyl, and then here's our nitrogen. And so we can see that hydrogen bonding occurs between that oxygen and that hydrogen atom. And as we'll see here, there's an important, we're not going to see this right now, we'll see when we talk about the structure of keratin and we look at an alpha helix helical wheel. We will see that there is essentially a repeat that we get pretty much around to a register that allows us to have that hydrogen bonding when we get about 3.5 turns around, 3.5 residues that we have around there. So here's one C alpha carbon, 2 C alpha carbons, 3 C alpha carbons, and there's the fourth one. So this is just about 3.5 residues to get around to where we have these atoms that can hydrogen bond. Okay, so zooming back out here, that's our alpha helix that we have here. So here's some important things. Highlighting common feature between both alpha helices and beta strands that interact to form beta sheets is that we're going to have hydrogen bonding between those backbone atoms. So here's how the hydrogen bonding is different with what we have with alpha helices. The hydrogen bonding that we see in alpha helices is going to be intrastrand. So intrastrand hydrogen bonding. And intrastrand meaning that it is within that strand. So we can see that up here. This hydrogen bonding is going to occur within this strand. Even though there might be another alpha helix nearby, it's not between those alpha helices, it's within the atoms within this single coil. And when we define what we have structurally with an alpha helix, we are going to have repeating, allowed phi and psi angles that create an alpha helix. Remember, that's those islands that we see on the Ramachandran plot. If you haven't watched the video about a Ramachandran plot, make sure that you do so that you can understand why there's certain allowed repeating angles that we have to create structure. So when we have repeating, allowed phi and psi angles, so those are those angles that are represented by the Ramachandran plot. So that's a Ramachandran plot island. One set of regular repeating allowed phi and psi angles is going to allow us to create an alpha helix. That entropically is unfavorable to freeze out all that molecular motion, so the enthalpic compensation is intrastrand hydrogen bonding. Okay, so one last thing I want to mention is when we look at alpha helices, I want you to think about the word coils. Okay, so these are coils. All right, let's move over and see what we see here with beta strands that interact to form beta sheets. So when we look at these beta strands right here, this is actually 2 beta strands. So this is beta strand number 2, and this is beta strand number 1. Okay, these guys aren't connected to one another, but let's take a look and see if we can see directionality here. Okay, find a blue nitrogen. So NC alpha carbonyl, NC alpha carbonyl, NC alpha carbonyl. So I can see that this strand is running in this direction. Let me look at the other strand. I can see that this strand, no, that doesn't go NC alpha carbonyl in that direction, NC alpha carbonyl, NC alpha carbonyl. So this runs in the opposite direction. So the N to C direction in my second strand is opposite. So this is what we call an anti-parallel beta sheet. Okay, so we have two beta strands running in opposite directions. Okay, so let's make two beta strands. Now, when I made my alpha helix, I wound it around my finger, and that had a certain repeating allowed bond angle to make a coil. It's going to be a completely different repeating allowed angle to make a beta strand, and that is going to be a zigzag. So I'm going to make a zigzag here, okay? So here is one beta strand. And as we're going to see when we look at a slide following this, we're going to see a different representation of where our side chains are. Remember, our side chains are the dangles that we have on our structure here. So here's one beta strand, and I'm going to make another. And we're going to see this in a minute. We call our beta strands, okay, zigzags. So here's another one, okay? And it is entropically unfavorable for me to fix these repeated allowed angles to create zigzags. So what I must do then is have hydrogen bonding. And between these strands is intermolecular hydrogen bonding. So I'm going to have hydrogen bonding between these strands, okay? And that's what we see here. We can see that our hydrogen bonding that we have here, let me go down here where I'm not kind of writing over something. This hydrogen bonding that we have here, so this hydrogen bonding is what provides that enthalpic compensation for freezing out structure. But in this case, it's going to be between strands. So let's go ahead and then contrast, zooming out here for a second, for alpha helices that was intrastrand hydrogen bonding, so within the structural element, but it was repeating allowed phi and psi angles that on that Ramachandran plot created an island that was a right-handed alpha helix. Let's go ahead and put that in here because this was a right-handed alpha helix. So I'm just going to talk about what we see then over on the other side here when we have beta strands. In this case, it's going to be interstrand hydrogen bonding. So inter meaning between, right? It's two separate beta strands that hydrogen bond together to make a beta sheet. This also reminds us that in order to make a beta sheet, we have to have at minimum two beta strands. Now you can have more than two beta strands that can be hydrogen bonded to one another to make a beta sheet, but at minimum you need two. And we can have, because of the orientation of our beta strands, either an anti-parallel beta strand or beta sheet, which is what we've got shown up above, or we could have this run in the same direction and it would be a parallel beta sheet. So we can have parallel or anti-parallel beta sheets. And so we have an anti-parallel one. How do I know that it's anti-parallel? Because I've got one strand running in the N to C direction this way, and then the other one is running in the opposite direction. So that's why it's an anti-parallel beta sheet. But what we're going to see down below this is a common theme. We are going to have repeating allowed phi and psi angles that are going to create, again, what we had over here, we had a Ramachandran plot island, okay? So that's going to create a different Ramachandran plot island because those angles are different. I had to make different angles in order to make a zigzag versus a coil, okay? So those different angles are now going to create beta strands, which are going to interact to make our beta sheets. And this repeating allowed phi and psi angle creating a different Ramachandran plot happens when we make zigzags. Okay? So that's how we form these secondary structural elements. The last piece to think about here is where the side chains are on these pieces. Okay? So when we think about side chain distribution, we want to think about where they are actually within each of these elements. We're going to talk about that on the left side of this plot here. And then we'll actually talk about where secondary or where side chains are when we talk about distribution within a protein. Okay, so kind of keeping with our theme here, everything that's alpha helix, we're going to have red. So over here, here is an alpha helix. Okay, so this is an alpha helix. And then we've got our beta sheet. And it's a little bit harder to see with these structures here. Let me just flip back just for a second over here. I didn't sort of show here, but it's probably worth mentioning these structures that we have over here on the right, they're the same thing, okay? Alpha helices and then beta sheets. But it's super easy to see things in a ball and stick model. But the problem with ball and stick models, or even the problem with what I have here, is it looks like there's a hole in the center of my alpha helix. Same thing with what I have here. But if we actually create this as a space-filling model, we'd see that there was not a hole in the center of an alpha helix. And that's a really important thing to remember, that sometimes our structural representations, to allow us to understand what's going on, create molecular space and atomic space that's not there. So space-filling models often are a way to make sure that we recognize that, but they're complicated because they're really dense and they're really busy, so sometimes it's hard to see the pieces that we want to be able to see. So let's flip back to these space-filling models to talk about side chain distribution. So side chain distribution for alpha helices. Okay, so I'm going to zoom in here. We've got kind of two different structures. I'm going to highlight that the gray that we see here, this is our backbone. So this is our backbone. So that's really what I have in red here. I don't have any side chains on this. Okay, so that gray represents our backbone, and we can see that there's not a hole down the middle of it. It's basically all full of space. And then everything that we see in purple and gold represent side chains. Okay, and in this representation here, the ones that are in purple happen to be hydrophilic. So hydrophilic side chains. And then the gold represent hydrophobic side chains. Okay, but the key that I want you to think about with the side chains that we have on an alpha helix is that the side chains are going to point out and then slightly down. And we can sort of see this here, okay? They point out, and then slightly down, out and then slightly down. Okay, so that's what we're going to see with our alpha helices side chains. Okay, so side chains, I want you to remember that they point out and down. Okay, and so sometimes, so out and down, and sometimes what we're going to see with our secondary structural elements like alpha helices and our beta sheets is that we are going to have a sidedness to where side chains are. So if we just look at this guy, right, we can see that there seems to be a collection of the nonpolar residues on one side of this alpha helix. And remember, alpha helix is kind of like a cylinder. It's full of stuff in the middle, but our side chains are spinning out around the outsides here. So we might have a side of our alpha helix that seems to be a little bit more nonpolar than another side. And what that's going to allow, remember, when we have our protein that kind of forms a tertiary structure, if I have a helix here that's going to be sort of orienting itself, it's likely to want to put its greasy parts towards the middle, away from the aqueous environment. Okay, so a little bit more quickly now, going through our beta sheet here. Here you can see still in the gray, we've got our backbone atoms. So that's all of these atoms here. And we can see our nonpolar residues and then our polar residues. So the key with where we see side chains in a beta sheet is that they alternate on the faces of our sheets. I'm going to go back to our other slide here in just a second to kind of show you this a little bit more clearly, because remember, space filling models are a little bit hard to see that with. But what we can even see with our handedness here is that there seems to be a face of our sheet that's a little bit greasier than this side. So again, we're going to see some of this distribution of side chains such that we see the hydrophobic effect in action. We're going to see things that want to be away from water kind of exclude themselves. Let me go back to this slide just really quick for a second to kind of highlight that we do see our side chains sort of alternate. So this represents, again, our peptide backbone, right? So here's our peptide backbone, and here's our R groups. So our R groups are alternating on opposite sides of this strand, okay? So as these strands come together, and if you want, you can even think of this zigzag, that this is going to be on the zig part, we're going to have a side chain, and on the zag part, we're going to have a side chain. So when I have these guys come together, what can happen is that I'm going to have some side chains face towards you and some face towards me. And what that creates with our beta sheet here is a face to the sheet, maybe a nonpolar face and a polar face to these secondary structural elements. All right, last piece here, and I know this video was a little bit on the longer side here, is when we start thinking about making an entire protein here and asking, where do residues go? Okay, what we're going to see here is that side chains basically are going to put themselves in positions that kind of satisfy the hydrophobic effect. Okay, so when we look at this, actually happens to be cytochrome C. This is going to be an enzyme that we're going to study later in the semester when we talk about the electron transport chain. One of the reasons I love showing this, just a little history side note here, this was actually a photo that was from my biochemistry textbook when I took biochemistry, and my goodness, I think this was probably 1992, okay? They didn't have, computational methods to easily make, space-filling models. So they had artists that actually drew this. So here you can see, here's the artist's name, and this was a, colored pencil drawing, an illustration of the three-dimensional structure of the protein cytochrome C. Okay, so a little history there. Little history aside, let's talk about what we're looking at with these two structural representations. This is actually the same protein, but different amino acids are colored depending on whether they're hydrophilic and green or hydrophobic and red. So if we take our hydrophobic residues, okay, so our hydrophobic residues are shown in red, and if we see where they tend to congregate, we see that they tend to be on the interior of the protein. So hydrophobic residues are going to be on the interior of the protein, and this, again, this is the hydrophobic effect in action. So hydrophobic effect in action. Hydrophobic effect is the tendency of nonpolar molecules to minimize their contact with water. So if we've got side chains that are greasy, they want to be on the interior of a protein. Okay? Contrast that with what we see with these green residues. So the green residues are hydrophilic. I don't know if there's two. I think there's just one L, hydrophilic. Okay? And these are going to be on the outside. Okay, and if we just look at this picture, we can see that, yes, all of these green residues that are on the outside here of our protein, okay? Water-loving stuff is fine being on the outside of a protein, okay? Now, you might see, oh wait, there's a couple that appear to be on the inside here, but they might have functional groups that allow them to do the chemistry that they're doing here. And actually, this is going to be a histidine residue that's going to help coordinate the heme that we see in the center of the cytochrome C. Okay, so I think that wraps up a longer video thinking about the different kinds of secondary structural elements we're going to observe. Alpha helices and then beta strands that interact to form beta sheets. So alpha helices are coils, and then our zigzags, which are our beta strands, which interact to form a beta sheet.